Before we dive in, let me ask all of you a question. A show of hands. How many of you have ever conducted an experiment? Wonderful, that's a lot of hands. Now, keep them raised if you have ever worked with living creatures in those experiments. I see a few hands still up there. For those of you in engineering or computer sciences, you might feel relieved because for us who work in biology or medicine, we face ethical dilemmas and moral decisions when innovation comes at the expense of sacrificing animals for experimentational innovation. But what if I told you that there is a technology that could transform all of this? A way to run healthcare experiments not on animals, but on tiny synthesized platforms, including chips. Of course, not electronic chips, but organs on chips. Together with miniature organs, they could mimic the human body's environment and could offer a powerful alternative to animal experiments. This is not science fiction. This is science in action. It is reshaping how we model diseases, how we develop drugs, and how we imagine the future of healthcare. And today, I invite all of you to step with me inside the fascinating intersection of biology and engineering. I want to explain to you about these technologies through the example of lung cancer research. Lung cancer, it's one of the most common cancers in the world, and tragically, it's one of the most lethal cancers in the world. In 2020 alone, more than 2.2 million people were diagnosed and nearly 1.8 million lives are lost. Studying it is challenging. It comes back often, it develops drug resistance fast, and for decades, researchers have relied on two main platforms, 2D cell cultures and animal models. Now, 2D cell culture is simple. You grow cells on a flat dish, and they spread out in a single layer. But here's the problem. Single-layer cells don't capture the complexity of real human tissues. Animal models would help with that, but they come with their own issues as well. Genetic differences, long development times, and of course, ethical concerns. So, where do we go from here? The answer lies in advanced 3D models, organoids and organs on chips. We enter a new frontier where miniaturized, lab-grown versions of human organs meet microchips that mimic the dynamic environment in the human body. Organoids are created by culturing stem cells with signal chemicals like cytokines outside living creatures, forming a 3D cell complex similar in structure and function to that of the target organ. They have stable genetic characteristics and are capable to survive for a long time. They have been utilized in different lung diseases research, such as asthma and even COVID-19. In lung cancer research, for example, the lung cancer organoid or LCO model has helped with modeling the 5 different subtypes of lung cancer in cancer biobanks, identifying gene mutations that affect cancers, and testing drug susceptibility. The other type of 3D model, organ on chips, comes in various sizes and shapes and could be built with silicon, medical plastics, and even filter paper. But they all contain hollow channels lined by living cells and tissues cultured under dynamic fluid flow. Some devices recreate organ-level structures like tissue-tissue and organ-organ interfaces, as well as provide relevant mechanical movement like breathing that could model organ physiology. It is more accurate and realistic compared to the 2D model and solves the ethical and other problems that animal models would bring. One of the earliest systems developed is, as you have guessed, lung on a chip. Researchers have utilized this technology to create lung cancer environments to study the development of lung cancer and clinical trials. In conclusion, Organoids and organ-on-chips can reproduce the environment of tumors inside bodies. They are used for cancer-related mechanism study and lung cancer drug developments. But lung cancer isn't just a problem of the lungs. It's a whole body disease. It doesn't stop at the lungs. It spreads and affects multiple organs, and the drugs we use to fight it doesn't act in isolation. It needs to be absorbed, metabolized, and sometimes they will harm other parts of the body due to side effects. That's why researchers are moving beyond the two basic models I have previously mentioned. By sometimes combining these models, we create organoids on chips. Features like mechanical stress and microenvironments make microorgan models behave more like real human tissues. And the vision doesn't stop here. Imagine a human on a chip. Multiple organ chips linked together, interacting just like our organs inside us. It's a glimpse of the future, a way to study diseases systematically as it truly is, and test therapies in a model that mirrors the complexity of the human body. For nearly a decade, organoids and organs on chips have shown incredible promises. For nearly a decade, organoids and organs on chips have shown incredible promises with the development of biomaterial sciences, tissue engineering, and AI. Some regulatory barriers concerning safety and privacy has also been lifted. The FDA in the US has lifted the strict requirements for animal testing to introduce these new models for evaluating drug safety and efficacy. Imagine this. Instead of testing new cancer drugs on animals and waiting months for results, scientists grow organoids and place them on chips that recreate blood flow and tissue mechanisms. Drugs can be tested directly on human-like systems, showing how they are absorbed, how they fight tumors, and whether they harm other organs. This approach speeds up drug development and brings us together closer to how the human body really works. The promise is swifter development and more ethical therapies designed with patients in mind. Apart from revolutionizing clinical trials and drug development methods, I'm also looking forward to their further application in personalized drug development. By using patient-derived cells, we can build disease models tailored to the individuals, test multiple therapies to find the most effective treatment with the fewest side effects for everyone. Organoids and organs on chips aren't just models of disease. They could be the models of the future of medical research. Thank you.